white rice, brown rice, arborio, and basmati rice, jasmine rice, long grain, short grain, and sushi rice, risotto and paella, there is sushi and mochi. We get to enjoy all these dishes and delicious flavors because we have rice, the number one staple food on earth. But have you ever wondered why we have these variety of rice? Why arborio rice is starchy, while basmati rice is soft and fluffy? Why one has a long grain and the other is a short grain? Well, it's all about genetics. So, welcome to the genetic basis of stuff and things. Rice domestication began around 9,000 years ago in Asia, where early humans started cultivating the wild rice, Oryza rufipogon. Through centuries of selective breeding, farmers chose plants with favored traits like larger grain and non-shattering seeds so they wouldn't fall off before harvest, and erect and short plants so they will be easy to grow and maintain. This process transformed wild rice into primarily Oryza sativa, the domesticated rice we eat today. Over time, this domestication spread, the rice becoming the staple food for over a third of the world's population. Traditional domestication was slow, but with modern tools like genetic editing, scientists can now accelerate the process, introducing desirable traits in just a few generations. However, over the years, the gene pool for new variation was lost with selective breeding, which limited our ability to improve cultivated rice growth further. And if we want to introduce new genetic diversity, we need to take some from the wild. Which is why searching for a suitable wild plant is a good starting point. For example, polyploid is a feature that scientists are interested in because it is associated with biomass, vitality, and adaptation to environmental changes, which are desirable features in domestication. Polyploidy in rice means that there are multiple copies of the 12 chromosomes it has. Sativa, for example, is a diploid, which means there are two copies totaling to 24 chromosomes, where tetraploid rice means there are 4 copies of each chromosome, totaling to 48 ones. Which is why you and his team have studied ways to domesticate wild tetraploid rice in 2021. The paper they published explained a new method that enables us to make wild rice ours to use and enjoy. Their method can be summarized into 4 key steps, starting with selection, sequencing, editing, and testing. The researchers began by screening different types of wild tetraploid rice from the gene banks and selected 28 candidates. These lines were from Oryza alta, Oryza grandiglimus, and Oryza latifolia. After that, they have examined the candidates and chose a line from Oryza alta with the best characteristics. They have also compared it to sativa in terms of domesticated related features, and finally they named it polyploidy 1 or PPR1 for short. The second step is sequencing the genome of PPR1. High-quality genome assembly was conducted and the details on the 48 chromosomes were collected, like the genes, RNA, and genetic polymorphisms were identified. They also analyzed how similar PPR1 to different Oryza lines. This analysis showed that PPR1 have abundant genetic information that are absent in the current cultivated rice. Then it's the genome editing step. Although PPR1 is a good candidate for domestication, it's still not the ideal crop yet, because of its non-domesticated features like height, broad long leaves, and small grain size. Yu and his team used modern genetic editing system to insert and correct important genes for domestication-related features. Finally, they would make and grow different PPR1 plants carrying different genes and examine the features of interest. For example, they have modified the SD1 gene, which is known as the semi-dwarf gene that played a huge part in the grain evolution during the 1960s, where we avoided famine by increasing the yields of grains. Mutation in this gene will produce plants with short but strong stalks so they wouldn't bend or fall off from stress. They respond well to nitrogen and have high integration rates. It would have been great if O alta and O sativa could be bred together so their superior traits could pass through future rice generations. But because one is a tetraploid and the other is a diploid, this process is difficult and requires scientific intervention to happen. However, with the genome map built by Yu and his team from PPR1, and the help of genome editing tools, we could take the good trait genes and insert them into our sativa or any existing crop to improve them. There are actually examples of the use of similar methods such as in wild tomatoes and ground cherries that enable us to domesticate wild crop faster to meet the increasing food demand for the increasing world population in the future. Such approach could be transformative for the agriculture industry because it could rapidly improve different crops, increase food security, it could allow us to produce new staple food, reduce farmer dependency on chemical and pesticide, make crops adaptable to different environments, and customize crops to suit our needs. So in the future, there might be new variety of food that we haven't seen before. So don't get surprised. Now you know how we got there. And remember, it's all about genetics.